despite being really complicated, actually one of the best understood organs in the body. But a lot of that has come from not just things like ECG or more modern imaging techniques. There's also a lot of experimentation that's happened in animals. And they use a whole different wide range of animals, which is a bit unfortunate, and there are some pushes to get rid of that. And of course, one of the ways to get rid of that is to use mathematical models instead. And that has quite a rich history as well. So starting in 1952 with Hodgkin-Huxley, which some people might have heard of because I think it's kind of big names, they've now come to full 3D realized models that include the mechanical effects as well as the electrophysiological effects. And they're operating as PDEs coupled with systems of lots of ODEs. We'll talk a bit more about them in a little bit. But yeah, it's very sophisticated, and they've actually got a lot of detail they've figured out through all this sort of extensive experimentation that they've done. Okay, so the way the heartbeat actually works is by waves of excitation that travel through the heart. And so we'll see a bit later how those work and how they can go wrong. But the basic story is when a cell detects signal, it gets excited, it depolarizes, and then that tells the calcium channels to let in calcium, and then calcium is a big intracellular signal that tells the cell to contract. So the actual pumping of the heart is governed by excitation, and this little picture here, we'll talk a bit more later, but that's basically just the voltage, which is kind of the level of excitation over time. So the cell gets a signal, jumps up, and then it returns back down to rest. Okay, so when things go wrong, instead of just getting a sort of well-controlled heartbeat that comes from a single point, travels through the heart, and causes everything to contract in the right pattern, instead what can happen is you can get other things creating signals. And the two common sort of big picture effects you get from that are either tachycardia, where you get a rapid heart rate, or you can have fibrillation, where it actually just completely goes wrong. And so if you get tachycardia and it's pumping too fast because the normal heart rate control is not there, you can sort of stress out the cells and it doesn't pump as well and things like that. But if you go to fibrillation, in the atria, it sort of sticks around with you and you survive, but it keeps triggering the ventricles, the main part of the heart, at the wrong times, and you lose all that sort of normal control. And if you actually get fibrillation in the ventricles, well then you just completely lose pumping function and you have minutes to live. So it is actually very serious, and indeed heart disease in various kinds, all summed up, is the #1 killer in the world. And it's actually quite difficult to treat because of this variability. So essentially we would just say everybody's heart is different, both in terms of the electrophysiology and the structure, the anatomy. And it actually makes things very complicated when you try and treat these arrhythmias that we just talked about, because one of the most common side effects when you give people antiarrhythmic drugs is arrhythmia. So you actually find this very complicated balance between all the different types of heart cells and all the different parts of the heart, and you can try and fix one part and something else goes wrong, or maybe it just doesn't work, and it's actually very difficult to treat with these drugs. And so we need to sort of work out how this variability is having an effect to improve that treatment. Okay, so at the cell level, we'll just take a picture here of what a cell sort of looks like when we think about it in terms of cardiac electrophysiology. Basically, we don't need to spend too long here, but you have many, many, many different kinds of currents, and these all open or close at different times. And together, the effect of all these currents closing and opening, letting in or out sodium, potassium, and calcium ions mainly, is what actually gives you that plot we showed before, where it gets excited and then it returns to rest. And if we were to actually write down, well, we grab one of the models here, the actual thing is just an ODE, so it doesn't look too complicated, except it's added up effect of many, many, many different currents, and all of those different currents are all switching on and off at a complicated fashion. So it does actually work out to be quite complicated. I haven't covered what all the currents are doing here, but just super briefly, you have an excitation spike when you get a signal. So a little bit of signal tells all the sodium channels to open. That causes your initial spike of excitation. Then it repolarizes a bit. Then the calcium channels open, and then it slowly takes time to repolarize. And that repolarization back down to the resting state is very important, because that stops it from getting excited again too quickly. And you don't want to get excited again too quickly, because that means that you're getting some sort of arrhythmic pattern, or you have more susceptibility to it anyway. Okay, so just super briefly, if we look at one of those ion channels, just to give an example, we won't go through them all because there's too many. This is the sodium channel, and so you have some conductance and then you have 3 different gating variables. So it actually has a sort of activation, which triggers when it reaches a certain voltage. And then you have 2 different ones that turn it off. One of them turns it off quickly to stop the spike from going too high at the start. And then one comes back slowly, which is what recovers the sort of being able to stimulate it again later. So together, those gating variables control the sort of dynamics of that channel. And then the way the maths actually works quite nicely, you can kind of go into some physical principles and figure it out. You have the Nernst potential, which is sort of the balance of potential that causes ion flow. So the ion flow is actually passive. It's just balancing to try and get to that Nernst potential. And that's just defined by the concentration of ions inside and outside of the cell with some physical constants there, you know, gas constant, temperature, things like that. And so then if we actually say, okay, well, we've got that one ODE, dV dt, but we actually have a lot more because there's ODEs for the actual concentrations of the ions. We also have the gating variables, so those M, H, and J, they all are turning on and off, and they don't do that instantly. They respond to try and get to some steady state value at a certain time, but the steady state value is actually changing with response to the voltage, so maybe we don't call it steady state. But it has something that it's trying to get to at a certain rate. And then you have these very complicated expressions for how those actually look. And those come, I mean, they look quite funny, and they are actually just empirical fits. So those have actually just come from looking a lot at what a single cell does when we look at just the sodium channel. What's going on, how quickly does it activate, deactivate, et cetera, et cetera, et cetera. Okay, so the model that we'll talk about for the first part is the Quartermunch, Ramirez, and Natel model. CRN, we'll call it from now on. And just to give you an idea of the sort of scope of these things, it's 21 ODEs altogether. So your 3 ion concentrations, a bunch of gating variables, and then the voltage itself. And it is decently stiff. I would call it stiff, but I've seen papers where it's like, oh, there's more stiff heart models out there and blah, blah, blah. And that's because, yeah, you have that very fast upstroke, and then you have a slow repolarization. So you have different time scales which make the ODE solution a little bit annoying, or at least you have to use small time steps. ODE 15S works fine, but it means it takes a little bit of time. And that becomes a big issue as we keep going. Yeah, so the Quartermunch model is calibrated, but the way they calibrate is they just go and look at a bunch of data and they say, well, everyone's different. We've got this physiological variability we're talking about, but we'll look at just sort of the average and we'll try and get that curve to match and we'll play around with it until we get something that looks good. And so that's what they go off and do, and they get a set of parameter values for that model. Which, fair enough, but it's actually sort of a lot more complicated than that. And one of the best ways to show that is the Quartermunch model, if you use the default parameters, gives you some sort of shape like this. I mean, I sort of just lazily drew that, but you see a big spike, and then you see a dome. That's for in the atria, an atrial cell, but depending on whose data you look at, some of them have spikes and domes, some of them are just a sort of triangle shape. And then other models that you look at, so not the Quartermunch, but sort of other competitors that are also looking at all this intense biophysical detail, that will show a triangle shape. And then it's like, well, who's right and who's wrong? And there's no answer to that because, well, it's variability. That's what we're trying to figure out and deal with. Okay, so how do we actually summarize that effect of, you know, this sort of excitation behavior? Instead of looking at what the actual curve does explicitly, we look at what these biomarkers do. And it's not the way some other parts of biology use the word biomarkers, so don't think too hard about that. But essentially, they are measures of the different important properties of that shape. So how high does it go? How quickly does it go up high? How long does it take to recover? Is obviously a big one, because as we said, if you re-stimulate too quickly, bad things start happening. So these things are the APD, the action potential duration. That's how long it takes to recover, and that is quite important. But so yeah, we have all these different things we can measure. So you get a signal when you do experiments, and you can go and collect all of those. But everyone is going to show different values for that. And even different cells collected from the same person will show different values for that. Okay, so there's just a picture here showing some data. It's just one way to visualize it because we have 7 biomarkers, so technically it's 7D space. But I've just picked out all the different 2D sort of pairwise scatter plots you could do. And the blue and the red are healthy hearts and then hearts that are under chronic atrial fibrillation. So when it is actually fibrillating in the atria, as I said, you survive, but it's not bad, it's not good. Has a lot of bad health effects. It actually causes the heart to try and adjust to that situation. So the properties of the cells change as well. They call it electrophysiological remodeling because they kind of actually change on a molecular, well, yeah, molecular or cellular level. And so what we can see is, yeah, even if you look at just the blue, you can see sort of big clouds of data. Because all these people, all these cells taken from different people, are all showing slightly different properties in terms of how they respond to that pulse. And then if you look at blue versus red, you start seeing bigger differences because some sort of remodeling has happened which has caused the cells from the unhealthy people to start behaving explicitly differently as opposed to just, you know, there's some variability there. But either way, the sort of big message here is, well, we've got this one model, the Quartermunch model or CRN model, and then we want to sort of model this. And to do that, we have to somehow get it to give these effects. So everyone has their own parameters. And the way we deal with that is using populations of models. So it's a quite recent technique, and it's been quite popular for dealing with variability specifically in heart models. I think there's a few reasons for that. I think it's one thing is that the heart models are quite complicated, and this technique is great when your models take a long time to run. But I don't see why this couldn't spread to a lot of other places, and a few bits and pieces of work around the place, including here at QUT, have been doing some stuff with POMS. But what you do is you just create a virtual population. So you've got some population of people that all have slightly different behaviors, and you try and capture that effect in a virtual population. So now you've got a bunch of models that all have their own parameters, and so then you've got a set of models which represents a population. And that might sound kind of like semantics, but the important thing is you can then go to, where's that button? You can then go to that population and say, well, let's see what happens when we try and compare the parameter values that get put in different groups. Or if we, let's say, let's try actually giving them a drug treatment and see how they respond. And so then what we capture is the fact of, well, yeah, well, maybe some of them have a good response to the treatment and some don't. Can we figure out which ones are doing what? So it's actually a very powerful tool for looking inside that variability and saying, okay, well, what's going on here? Why are these people responding well? Why are these people not? Okay, but the issue is, if we've got some data, so we've got that biomarker data, the blue and the red dots from before, how do we actually select the populations of models? How do we select those groups of parameter values that give us that sort of overall effect? And you could say, well, maybe you try and match every single individual dot, but that gives you a very fixed population size for one thing. It might not be easy to find the perfect sort of values that get you close. And even then, the models are the case that you could find different combinations of parameters that all actually get you to the same point. So how do you know which one to pick? So it's actually better if you use a sort of big picture approach and try and get a big picture population that represents on a sort of global scale what the overall variability looks like. It actually helps regularize things. Okay, so what we're going to do is we're going to say, okay, that CRN model, so 21 ODEs, we're just going to think of it now as you give me a set of parameters, and the parameters that we're varying are the current strengths, and it will give you out that curve that we just saw, and we can say, okay, so what are the biomarkers? We'll just measure them from that curve. It's pretty easy to code up something that does that part. And so what we want to do is say, okay, yeah, can we find groups of theta that will give us a bunch of Bs that look roughly like that variability we saw in the overall population? And the thing to keep in mind is that here we've got, in our case, it was 11 parameters we're varying here, and that model is 21 heavily nonlinear ODEs. So that relationship encoded there by the capital M is quite complicated. So how are we going to deal with this? And the literature kind of says, let's not. The literature says, let's actually just draw a big box. So we will select any model that looks physiologically reasonable. What's reasonable mean? It means it's in the limits. And so if you have a 2D example like here, it means take, if the x's are our data, take the lowest part and the highest value, and then you can actually just draw a big rectangle around it, and that's your region of things that you will accept. So you just keep running your model for lots of different tries, and anything that falls inside that dotted line, you say, yep, okay, that's going to be part of my population. And obviously, we would prefer something that looks more like the blue. Because if your model tends to give you values over here, I mean, you're just putting parameters into a very complicated model, so you don't know what it's going to give you. It might tend to give you things here, and we want them actually here. And you might be able to sort of picture, we've got 7 biomarkers, not 2. So if we think about that in 7D space, drawing a sort of big hyper-rectangle in 7D space is only going to leave more gaps everywhere where we shouldn't be putting models. So we really want to try and select things that will be at the right points. How do we do that? Well, let's try and do a bit better than what we're calling here range calibration, just looking at the ranges. And the way we do that is sort of a Bayesian approach. And so I don't want to go sort of too heavy on this stuff, but the overall sort of framework that the Bayesians operate from is you've got some sort of prior guess at what you think's going on. And for us, we don't really care. Let's just say that's just flat. So it could be anything within some certain range. And then here you have a likelihood, which is the probability that you would have seen that data, so that set of biomarker values, given those parameters. And immediately the whole thing kind of breaks down. Because usually when you're doing this, you've got a set of data and you're trying to find parameters that will look like that data. But here, if you pick one set of parameters, you might find something that looks like one dot, but you're not going to find something that looks like 252 different dots at once. Because, well, firstly, you're only getting one answer. So there's no way to actually say the probability that we would have seen our data, 252 answers for one set of parameter values. So unless you do some playing around with adding in noise and then making that noise large, which is probably a whole other approach you might try, you can't really put this in the Bayesian framework naturally, but we can borrow some techniques. So the technique that we use is sequential Monte Carlo. And what we're doing with that, essentially you can think of MCMC, so Markov chain Monte Carlo or sequential Monte Carlo, as methods for exploring a parameter space. And you're trying to find high density regions. So in the probabilistic context, it's high probability. For us, it's high density of dots. And the way we turn that into probability is just by putting a Gaussian at all those different little dots. And you add up all those Gaussians and you get a sort of smooth approximation of probability to what that variability looked like. So putting all those Gaussians on gives us a probability distribution, which is this P of B here, B for the biomarkers. And what you do is you just pick a bunch of particles, put them all sort of randomly through the space. But then each time you go through this loop, you resample them according to their weights. So if they're in a high density region, it's more likely that more particles will end up there when you do the resample. And then what happens is you just jiggle them around a little bit. So you have a movement step here which just says, okay, move all my particles. Because when you do the resampling, let's say you had a high weight, well then maybe 4 particles all weighted onto that one because you just selected according to the weights. So high weights will get a bunch of duplicates coming onto them. And what we don't want is, oh, we started with 500 particles and now we've got 10 because they've all sort of duplicated onto each other. So every time after you do that resample step, you do a jiggle step, and that's just where you try all these other different locations, usually nearby, for those points, and then you only accept those trial jiggles at a certain probability that preserves the probability distribution. So that's essentially what MCMC is doing, and then the sequential Monte Carlo, the extra step is just we have this gamma in here. I mean, I'm being sort of vague about it, but the big difference is when you've got this gamma, you start gamma equals 0, and so if you think about probability to the power 0, that's just 1 everywhere. So you're starting with just flat space, and then as you increase gamma up to 1, you're slowly reaching the true p to the 1. So you're starting with a very flat probability distribution and slowly introducing the complexity to try and capture the actual probability distribution you're trying to recover, which in our case is a bunch of particles that look near that variability. But we actually sort of cheated a little bit because here we're moving in terms of theta, because we're picking different theta values, different parameter values to put into our model. But we're doing all our calculations in terms of our probabilities, in terms of the biomarkers. There's actually a probability transform in there we haven't included at all. And there's no real way to figure it out because that is encoded by that horrible nonlinear ODE model. But by doing this, we're still weighting according to high values of that probability. So we're still weighting towards high density areas in the parameter space. So we can go away and do that. We just need to do a bit extra after that. And we'll see the effects of that in a second. But the way that we did a sort of extra step was we said, okay, let's say we had 200 data points. Maybe that means we want a population of around 200, or maybe we want, say, 500. But what we're going to do is pick 2,000 particles, put them all in there. They'll all sort of be jiggled around and converge towards something that looks kind of right. And then we're going to pick the best ones, essentially. And we pick them in a way that maximizes the agreement between distributions. So you're not picking individual particles that are close to the original dots. You're looking at a sort of big picture sense. Here's the distribution of our data. Here's the distribution of our particles. Do they look similar? Yes, no. Well, let's try getting rid of that particle, or we're putting that one back in. And so we take that 2,000, cut it down to 200 or 500 or whatever it is, in a way where we keep saying, yep, we'll try different things, and if it's looking better, we'll accept it. If it's not, we will potentially accept it at a certain reduced probability. And simulated annealing, essentially what you're doing is saying, okay, let's just try lots of different combinations, but to try and find better ones, we're definitely going to accept the ones that are good, but we're going to accept some that are bad tries as well, because that will help us not get stuck in a local minima. Because if you just only accept good moves, you might end up stuck in a little dip without finding the true best answer. So that approach is just a way of sort of avoiding those local minima while still trying to find the optimum subpopulation from the big group of 2,000 that gives us the best fit. And I don't know how well I'm explaining that, but we'll see in the pictures what's going on. So the white is the data. The red is what happens if you use range calibration. So that's sort of what the literature had been doing. And you notice these big sort of artificial rectangles are appearing. These funny corners here that you really don't want. You're putting lots of models there that shouldn't be there. And this is, again, visualizing in 2D, so it's probably worse in 7D. But what we see with the blue, so that's using the sequential Monte Carlo, is yes, we are getting more in the right place. But there are still some sort of, like there's a big chunk of blue down here we don't want. And it's sort of a bit too fuzzy. We'd like to sort of refine that better. So that's where we use the simulated annealing to pick subpopulations. And so the red is now just what was blue on the previous. And now we're selecting from the red sort of the best ones, which are the light blue. And that does pretty great in all cases except one of the biomarkers, this upstroke velocity here. Like for example, this has just completely failed. And that's because the variability we put in our model isn't enough to actually explain the data. So there's other variability in there that we didn't introduce. And that was, of course, a reviewer comment. And we went away and said, well, if you introduce this variability as well, it starts doing a lot better there. So that's actually, yeah. It's showing that depending on what variability you put into this process, you will get different answers. And you can see, is that enough to explain the data? And if it's not, you've identified, hey, there's more variability there. So the technique also works to actually say, well, are my assumptions about the variability in this physiological system correct or not? OK. But so the other sort of conclusions, that was one of them I just sort of gave you there. The other conclusions that we get from this work is if we use that sort of data density calibration, so we're getting, the nice light blue here instead of the original red, we do a lot better at first reflecting the data. I think we can agree with that. But we also then go and show why it's important. And so what we show is that if you have two populations, which we did remember, we had the blue and the red data right back, the first time I showed that stuff. So these pictures are just for the blue data, but the red one does pretty much the same thing. It's just different data. If you then go and look at the difference in parameters you selected for the blue and the parameters you selected for the red, it actually picks up a lot of the ionic effects that are known to be associated with that electrophysiological remodeling. And so just by looking at the big picture data that's easy to measure, which is the biomarker data, so it's sort of easy to collect compared to doing all these individual cell, individual ion channel studies, we've been able to pick up most of the changes that electrophysiologically underlie that difference in the data. And that's a big deal because then you could also go ahead and say, all right, well then let's get a big group of people, might even be the same people we had here, but then we could say, well, let's check according to age. And so we say, people over 60 here, under 60 here. Can we see a difference in electrophysiological properties between these groups? Another big one is actually biological sex is a big sort of discriminant for different sort of things. Like there's different risks of arrhythmia depending on whether you're a girl or a guy. So that's another group you could say, yep, let's cut them up. And then say, OK, can we see any differences electrophysiologically, sort of the under the hood parameters that we can't see when we just go and measure the biomarkers? Can we identify the differences that sort of underpin that variability between those two groups? And then the other point was, yeah, as I was saying, if you get an answer and it doesn't quite match what you've done, so you've put tons of variability in, the model still doesn't want to predict the data, that suggests that you probably have other variability that you didn't include. And in our case, it was a time constant. So those gating variables have time constants associated with how long they take to open and close. And if you introduce some variability into there as well, then we started seeing this funny line, we were able to sort of, properly fill that out to get the data. But just varying current conductances, you don't get that. So yeah, it's actually a technique to sort of look for questions like that as well. Just the last part about the cell level stuff is, as I said before, once you've got a sort of virtual population made up of all these models, what you can do is then say, well, okay, let's try and give them a drug treatment. So as I said about how long it takes to sort of recover back down to rest is a measure of how risky it is. And so if it's recovering too quickly, that's bad. And one of the changes, according to that remodeling, that happens when you have atrial fibrillation is it gets shorter. So we want to use drug treatments to make it longer. So we can take all of our red lines, and those are just all of the curves we get from simulating our population of models. So we've got, 200 and something there. We just plot them all. And you get the variability manifesting in all these different measurements. And then we say, okay, we'll give those 200 different virtual people a drug treatment that blocks one of the ion channels, so changes the electrophysiological properties. What happens? And we see in almost all cases, you get the proper prolonging effect. So you get basically the black versus the dotted black. The drug treatment on the whole is doing the right thing. But then you see, hang on, there's a few here that are suddenly repolarizing extremely fast to the point where we say, well, maybe we put too much variability in the model because that doesn't seem physiologically reasonable. But regardless of how accurate we call that, because it might be very extreme parameter values that do it, it does tell us, if we then go look at, okay, well, which parameter value had to be extreme to make that happen? And so we can then identify, okay, in this case, these ones were associated with a very low value, I think it was, of the sodium-potassium pump. So you can actually identify what puts people at risk of adverse reactions to drug treatments. Okay, so that's sort of the science advances work. It was all about, how do we actually do a better job of calibrating to a distribution instead of just sort of the box. And a few other people started doing that around about the same time we had put ours out in review. But one of the important things we did was we said, okay, well, why is it important? So other people kind of said, well, you could do this statistical method and you can kind of do it. But we said, okay, we can also show why it matters. So that's that work. Then what we've done is gone to the tissue level, which is a lot more interesting, but also a lot harder. And so at the tissue level, now what we're talking about is, well, in our case, we're going to use just 2D sort of layers or slabs. I think I was saying slab, and I was told not to say slab, but I said slab. And so we've got now spatiotemporal spread of excitation. So instead of just looking at what a single cell does when it receives stimulus, we're going to let that stimulus spread from cell to cell. And you might guess that's going to give you traveling waves because you've got a sort of spike of activation that leaks into the next cell, which then makes that one spike, and so on and so forth. And just to sort of give a little bit more background, I won't go into this 3V model, but what is actually happening when you go to tissue in 2D or 3D or 1D, here you have an I-ion term, so it's just diffusive spread of the signal, rate of change, so just a kind of normal diffusion PDE, except here your reaction term is that CRN model, or in this case it's the 3V model, which is greatly simplified just to sort of help with computational time. But that would, if you were going to do a sort of fully biophysiological detail simulation in tissue, be 21 ODEs hiding inside that I-ion that sort of define what that I-ion does. And even if you use the 3V model, when you put this into tissue, it still actually requires quite an amount of significant computational time because you have that very rapid upstroke. And if you want to properly resolve where the traveling wave is moving when it's sort of excited so rapidly, it means that you need very fine spatial and time discretizations. So it still takes you quite a while to simulate these. Okay, so I'm just going to now go through a bunch of pictures that will show the sort of things that we're talking about when we talk about healthy excitation and tachycardia and things like that. And I think they're pretty cool anyway. So that's sort of the boring one, because that's the healthy one. So if we pretend this is the node that either triggers the heartbeat or triggers the heartbeat that came from the atria into the ventricles, it just excites the tissue and the excitation spreads through the tissue. So fair enough. It doesn't do anything funny there. That's great. But what can happen is if you have two stimuli that interact with each other in a funny way, and here I'm just calling it artificially inducing the spiral because I'm not going to really go into how precisely we generate it, but in the heart you can have sort of what they call ectopic beats. So you get a little bit of excitation that comes from somewhere else, and then that interacts with the normal wave traveling through the heart, and it can sometimes interact in very funny and dangerous ways. And so you can generate things like this. And so now you might look at that and say, well, that doesn't look dangerous. But what that actually means is instead of the heart being excited by one point down here at a regular rate that the body has control over, and according to sort of hormone responses, it says, oh, I'm exercising, I need to beat faster or whatever. Instead, you've just got this annoying little spiral of excitation sitting somewhere in your heart saying, go, go, go. And it's going too fast. Like, that's why it's tachycardia. So you don't want these. They're bad. But you can have several varieties of them. So there's actually a range of different shapes that you can see. But just by changing the electrophysiological properties in that model, we still just get what we'd call a little rotor saying go, 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 go. But now it's triggering the heart at a different rate and it's wandering all over the place. And that's important both in terms of, okay, well, what rate is it triggering, but also where's it going? Because the little one that's just rotating in one spot, it's quite stable. It's not going to run into a wall. It's not going to run into other things as easily as something which is sort of wandering all over the place. So a measure of how likely you are to see this also depends actually on, well, how far is one of these things going to wander around if one was to arise? Because it might just run into the wall and die. And in that case, you're safe because it's not sticking around bothering you. And the other thing that can go wrong for different kinds of parameters is spontaneous breakup. So nothing in that simulation actually was different to the previous ones of the spiral except parameter values. So we haven't sort of triggered some extra stimulus in there. It's just a natural sort of way that the wave front interacts with the wave back. And that one's worth probably looking at again, where we see, yep, it just runs into itself in a funny way, splits up, and suddenly you have spirals everywhere. And so that is one way, it's probably the predominant way that you'll get fibrillation in the heart. There's just some contrasting theories about whether this happens at the rotor tip or far away. There's a few sort of complexities there. But that's the basic story. Based on what parameters you put into this model, we can either see a stable spiral. We can see a spiral that wanders around everywhere and probably just ran into a wall and died, because we have used quite a big slab of tissue here to sort of see the effects. Or you might see it, yep, just breaks up into chaos, bad fibrillation. So, but before we actually jump into looking at how these different parameter values affect the sort of excitation pattern that you see here, one thing that people have done a lot of looking at is a sort of big picture measure of how bad these arrhythmias are or how likely they are to be sustained or to be induced in the first place. And that's the concept of excitation wavelength. And so it's a measure of how fast the excitation spreads multiplied by how long it takes for the tissue to become excitable again. And the basic idea, as you can see here with this little sort of ring image, is that if you have some excitation that's trying to travel around in a circle, in this case it's around an obstacle, but it doesn't have to be, as it moves around that circle, it has to keep finding new excitable tissue to sort of invade into. And so the faster that it moves, or the longer that it takes for the tissue to recover, that's actually going to then increase the chance that it runs into itself. So if you look at the picture there, you've got sort of the red, which is the recovering tissue, the blue, which is the excitable tissue. There's a little yellow sliver there, which is the sort of excitation wavefront. That's where it's being excited right at this very moment. And what happens is that yellow part's going to move through the blue, and if it goes too fast, or if the red sticks around for too long, it'll run into the back of the red. And then the excitation is going to disappear because there's no excitable tissue. So this is sort of a big picture way of looking at how dangerous the arrhythmias are, or how likely they are to sort of stick around, because it's a measure of how much, what chances there are that it will run into itself and basically annihilate itself, and then the excitation will be gone and the heart will go back to normal. And we're going to try and look past that, however. Okay, but so to look past the wavelength, we're going to be changing parameters which are at the cell level. So these are things like the strength of the currents and their time constants that control, how fast they switch on and off. And that's our theta. But then we have now, as the sort of outputs of our model, the biomarkers B, and they are measures which come from those excitation patterns we just saw in the videos that measure how bad the arrhythmia is or how likely it is to stick around in a sort of very quantitative sense. So some of the measures that we were using were, we have the maximum distance, so that's the rotor tip itself, how far does it actually move around, and what's the maximum distance between any 2 points on its path. And so if you have a little one that's spinning in a small circle, then that's going to have a small distance. If you have one that wanders around everywhere, then that will have a big distance. And that's important because that controls sort of how much tissue you need for this thing to sustain itself. In the heart, it might run into a boundary and then just die out, and then you go back to a normal, healthy heartbeat. And so the sort of the amount that it wanders around is actually very important. We also then have the dominant frequency, and so that's a measure of how quickly the heart is being stimulated. And we measure that by actually taking a lot of probes in the heart, and or in our simulated tissue, and they will measure what the voltage trace was at their individual points. And so then we can then look at how frequently those individual points all being stimulated. And we end up then, if we put that into the frequency spectrum, when we get the power spectrum, we can say, okay, where was the dominant peak of this? That's where it's being stimulated sort of the most regularly. That's the closest thing we have to a sort of measure of the frequency of how fast it's being stimulated. And that's relevant because that's then how quickly is the heart being driven or triggered by these little rotors. And they are always going to be doing it faster than what the normal heart wants to beat. So that's a measure of sort of how badly are these arrhythmias actually stressing out the heart and causing it to beat too quickly. And then the last thing that we measure are the organization indices. And the important one in particular is the one that tells us whether we've got chaos or whether we've got a regular spiral exciting the heart at some sort of regular pattern. And that is also calculated using that sort of power spectrum that we get from these little traces from all these little probes. And we're basically saying, yeah, well, is it being stimulated at a regular rate, or is it being stimulated at sort of just complete chaos? And that's important for telling us, yeah, well, do we have fibrillation or do we have a spiral? Because it's obvious when you look at it which one you have. But when you want to run this model a bunch of times, you don't want to go check every individual one. And especially when we later go and do the emulation where we run it many, many, many times, we need some sort of automated way of calculating that. And that's what these biomarkers are doing for us. Okay, so we have those cell level parameters theta. We put them in and we get the biomarkers out. So we have 8 variables in this case, the cell level parameters. And that's actually quite a lot because we're trying to explore fully what happens as you vary those parameters. How do the biomarkers depend on that variability? And so with 8 parameters, that means you've got an 8-dimensional parameter space. And how are you going to explore that? By running your model lots and lots of times. You would need so many runs to sort of cover that whole space. And in this case, that's especially bad because every time we want to do one of those runs, we're running one of those full 2D tissue simulations. And the tissue level simulations are especially costly because you have an excitation dynamic which happens very quickly, and it has to be sort of resolved very, very accurately. And you have that really strong nonlinearity of that initial upstroke where it excites very, very quickly. So we need very high temporal and spatial resolution in terms of our sort of numerical techniques that will go away and solve that to actually get accurate results. So the big picture story there is you've got many, many runs that you want to do to fully explore this huge parameter space, but each of them is very costly. And those two things don't agree with each other, and that's why we need to turn to a technique, in this case, emulation. Okay, so emulation is techniques that actually sort of replace the model that takes a long time to run with something that takes almost no time to run. How do we do that? Well, we want to just look at the biomarker, so like our prediction from what the model does. We want to fill out that response surface. So we want to fill out that surface using just a little bit of data, and then by filling that out, we're going to say, well, if we pick a different parameter value, yeah, I think it would be there. And so if we fill out that surface correctly, we don't have to run the big complicated model anymore. Is it easy to do that in 8D? Not necessarily, but we do a decent job, as we will see. And basically this slide is just saying if you want to get a good resolution of that surface, you want to choose points that are sort of evenly spread. But being conscious of time, let's just say that. Okay. So Gaussian processes are one of the most popular techniques for emulation. And emulation is very popular in climate sciences and also in engineering. They use it a lot. And I'll just say that it's also known as kriging. So if you've heard of that, that is Gaussian process regression or emulation, because you're just filling out a surface. So it's still regression or emulation, whatever you want to call it. And the advantages of Gaussian process is it's non-parametric. What that means for non-stats people is it means that you don't need to specify some parametric form of what you think your relationship is. So you're not saying, oh, it's linear, let's try and get the best linear fit, or whatever shape it is. You're saying, okay, I've got some weird looking surface, but I can fit it. And they perform very well. That's why I guess they're one of the most popular techniques. And they give you a measure of uncertainty as well. So they say, yeah, I think you've got this level of error in my prediction here. The bad things are they're kind of hard to train, and the formula that you get to actually make the predictions are not easy to interpret. Obviously, linear regression gives you a nice linear formula it's easy to look at and think about. This, not so much. You have some weird matrix inverse product thing. But yeah, the only point that we'll have to say, though, is with Gaussian processes, they assume smoothness in your data. And we don't necessarily have that because we're going to have some parameter value where it's doing a nice spiral and then suddenly it's fibrillating. And if it's fibrillating, we can't even measure a rotor trajectory anymore. So if we can't measure a rotor trajectory, one of our biomarkers is just going to take a null value. It's just going to be -1 or NA or whatever you want to call it. So in that case, we're going to have sharp transitions in our parameter space that the response surface has to somehow capture. And so we actually need to cut up our parameter space. And we're going to create separate emulators for all these different regions. And this is just a quick slide that shows if you don't do this and you just use one GP across the whole parameter space, when you have a bifurcation, it completely messes up, both because it can't necessarily detect that you have a different behavior here. So the covariance properties, it trains on this because there was more of this, and then it goes, yeah, it's mostly flat and easy moving, and then it just tries to do a flat and easy moving through here and completely fails. So obviously we want to draw that line down the middle and then get the blue line. How do we do that? We can either do a supervised approach, which is quite new, I think, and that's what we've done, and it's when you actually have values in your data that say this is this type or this is that type. And so we were able to go, yeah, we can tell that's fibrillating. We can tell that's a spiral. We can tell nothing happened with that one. So we have nice classes there we can use. And we're going to do different emulators for each different class. Because that class will be, yeah, okay, we can measure rotor tips for this one. We can't for that one. So that boundary between those two classes of behavior will be where we want to draw that boundary anyway. The other approach is unsupervised, where you don't know what the classes are and you kind of have to learn them by looking at the data. That's harder and doesn't work as well when you do have this available, and let's not talk about it anymore because time. Okay, so we won't talk too much about the actual methods we use, but support vector machines is the machine learning approach that we use to draw those boundaries in. So we generate 2,000 points, I think it was, and then these dots that are coloured in here are the ones where we're trying to make predictions. So they weren't involved in training, but we look at them and say, did they come out the right colour? For the most part, they did. So now our different classes of behaviour are predicted by this support vector machine. Why is that important? Well, then it means when we go to the emulator, it will tell us which emulation surface to use. Which Gaussian process regression do we pick? Because you don't want to pick the wrong one, because if you're in this region where no rotors develop, well then all your biomarkers are probably just like null. They're just -1. And if you want to get a nice sharp transition between these two, instead of a smooth thing that kind of messes it up, then you need to split up these regions, which is what we're doing. Okay, and so here's just an example surface of what we get. In 2D, the problem's not too bad, and we have quite enough data to kind of fill out the surface. So all the red data is the training data. White are the predictions, which kind of sit on the surface too, so that's great. And we've filled out that surface. So now you can go away and put in whatever combination of those two parameters you want, and it will tell you what it thinks that biomarker will be, and it does quite well. And if we look at the sort of results as just in terms of, okay, that's the true, that's the emulated guess, we're sort of sitting right on the line. We get a little bit of error, but it's doing very well, and it does very much better than if you don't do this partitioning thing where you split up the different regions. So that's great. But when you go to 8D, it's a little bit worse, but we still are actually picking up the correct sort of trends. We are saying, yes, we are sitting along this line. So we are predicting things that look roughly like the, yeah? Okay, yeah, this is close. Yeah, so we're picking things that look, pretty good. And if we're picking things that look good, then that means we can go away and use this emulator now to start doing some analysis in this 8D parameter space. And the way we do that is using the main effects is what we did. I mean, there would be other options. And you could actually draw maps like that 2D picture back here. Obviously, you can't draw that in 8D, but you can take like slices through 8D and do some interesting things with that. Anyway, what we're going to do is we're going to calculate the main effects. So what you do is you say, well, what is the dependence on one parameter when all of these different parameters are all varying? So how do you deal with that? Well, you just integrate over all the other parameters. So the effect of that parameter kind of gets averaged out. And if you look back at the 2D one, Okay, so you're saying, okay, I'm going to move along this axis, and I'm going to average over whatever I see vertically, and that will give me a rough effect of what this parameter is doing. It won't tell you the full story, but it tells you a sort of big picture effect. And you can do that in 8D as well very easily. The problem is to do this integral, or to do a multicolor approximation of an integral, you need lots and lots of points. But we have an emulator that runs in no time at all, so we can use those approximated guesses to actually perform this numerical integration, and that gives us those main effect curves. As I vary this parameter, what is it doing to this biomarker? And that's what we see here. And to talk about these in a lot of detail would involve doing more electrophysiology, and we don't have time. But even these funny things like this here, which looks like it's just giving mess, that's actually because we're measuring the distance. And the distance, you have spirals, and then you have little flower shapes, and then you have traveling helixes that actually just wander through the thing. So as you vary through all those different shapes, and they actually were in one of the earlier pictures, but I forgot to talk about it and we don't have time, you actually do see this sort of complicated dependence on the size taken, because as you go through the different regimes, the size starts going back up, and then it goes back down. And so that is actually a sort of reasonable effect. And the other things that are interesting is, yeah, well, do I get a big effect or do I get a small effect for this individual parameter, even after I take into account variability in all the others, which is what we're doing with these main effects. And let's not talk about them else too much, except here we have the GSO and the GSI. So in the model, that's a potassium current and a calcium current. One goes in of the cell, one goes out from the cell. So one pushes the potential up, one pushes the potential down. So you kind of expect opposite effects for them. Sure enough, in terms of the duration, in terms of the distance, in terms of the frequency, that's what they're doing. And that's basically saying, oh, I've got, it takes longer to recover. So it means I have to rotate slower. Fair enough. And then if I reverse, if I do the other current bigger, well, then it takes less time to recover, so it rotates faster. So that makes sense. But then we come over here and we say, hang on, it's doing the same thing in response to both of those. So what that actually means is probably that time it takes to recover, the APD or the refractory period that's strongly related, is not actually what's controlling that behavior at all. So we can't just look at the wavelength. We actually start getting different effects for the different components that make up that wavelength we're talking about. So in terms of excitability, which is kind of the velocity, we see that, okay, that controls the shape of rotor paths, and it also controls the risks of fibrillation, but in a sort of complex fashion, because you've got this sort of critical value where you're most at risk. And then in terms of the refractoriness, that controls how bad the tachycardia is, how fast does it rotate. But it has a very funny effect in terms of fibrillation, as we just talked about. And the last thing that we can talk about then is how do we try and target these things for treatment. And so for tachycardia, if we want it to rotate slower and be more likely to run into a wall and die, then we want to use refractoriness, so drugs that target that. On the other hand, if we want to target specifically that breakup effect, then we start looking at the sodium and calcium currents, so excitability and then also rate dependence. But I'm just kind of throwing buzzwords at you now without explaining cardiac electrophysiology. And that's pretty much everything. Future work, maybe we don't care about too much because we're out of time. Probably questions are more important, but that's some things that we are sort of working on or could work on. Okay, so thanks very much. And yeah, any questions?